right, so I'm going to change focus quite a bit here from clinical trials to probably some of the most basic research you guys are going to see today. So I also don't have any conflicts of interest, but at some point I hope one of these technologies takes off and I'll have enough money to say that I actually have a conflict of interest. So the motivation behind the work that I do is based on the fact that most healthcare-associated infections, about half of them, are attributed to indwelling medical devices. The ones that we think of, as emergency physicians would be central lines, endotracheal tubes, Foley catheters, but it could be, we were just talking about breast implants or expanders, prosthetic joints. Several different things are sort of involved in this effort. But basically, we've been working on this for years and we haven't really moved the mark. It's still the #1 cause of healthcare-associated bloodstream infection. So this is about biofilms. The reason why these things are so hard to treat is because bacteria form a biofilm on this device. Biofilms are actually important in lots of places other than healthcare. This is a biofilm that came out of a Coast Guard cutter biodiesel fuel line. So essentially, the boat stopped because it was full of a biofilm. It was running on biodiesel. This is a biofilm that's growing on a VP shunt that we extracted from a patient here. And you can see actually the little balls there. That's Staph aureus growing in a fibrin clot on a VP shunt. Here's a couple of pictures. One of them is a rabbit model that we've created of central line infection, and on the right is actually a human dialysis catheter that was infected with Staph aureus that we extracted and isolated as well. The problem is biofilms, they provide protection from the host defense, so they prevent diffusion of the humoral factors that get in there. They kill the neutrophils that try to attack it. They provide protection from antibiotics, which is different than resistance. They actually restrict diffusion of the antibiotic. They reduce the metabolic activity of the bacterium to prevent the antibiotic from getting in. And it actually degrades the small molecules of antibiotics. And then lastly, they have persister cells, which are like the stem cells of a biofilm. So this is the problem. So I'm going to talk about sort of 3 technologies that we're interested in trying to solve this problem. First one is the zinc oxide nanoparticles, which some of you may have seen was one of our more highlighted publications recently. We'll talk about a project that was originally John Younger's brainchild about using heat to treat biofilms in place so that you don't have to remove the device. And the last one that'll fit in with Alex Rogers' project a little bit, which is this rapid detection of bacteremia using a PCR technique. So this started with a bet. Gentleman, Nick Kotoff in chemical engineering, bet me $20 that this material he had, which is a polyurethane, nano-textured surface, nothing would stick to. No bacteria, it was impermeable, it was the Teflon of materials. So actually Bill Barsan was instrumental in helping me to get the support to build this device, which is essentially a way to look at bacterial adhesion to a device in real time. And so what you have here is a microscope that's sort of looking at the bacteria, and the bacteria glow in the dark green. And what you'll see here is the bacteria that are flowing past the surface, and anytime you see a little dot appear, that's a bacteria that's stuck to the surface. This is the actual movie of the material that he bet me $20 on. This material looks like this under a microscope. It's this sort of nano-textured polyurethane, which by all purposes looks like nothing would stick to it, but you actually see the bacteria climb down inside these little pegs and bent them over on top of themselves to protect themselves. So it's a very complicated problem. Trying to win his money back, he gave me these zinc oxide nanoparticles. And these are the ones that are a little more exciting. So zinc oxide, it's sunscreen, right? It's the most common ingredient in sunscreen. When you make it at this sort of nanoparticle size, these are very tiny, 26 nanometers, 4 nanometers, 25 nanometers, very, very small. To give you an idea of how small that is, right? It's smaller than an influenza virus. It's about the size of an albumin molecule or smaller. So these are really, really tiny. And what's interesting about them is they're antibacterial. So what we did is we coated these sort of pegs on this lid, and the nice part about this, you can do 96 simultaneous experiments in a plate like this, but essentially you can dip these positively charged particles in a solution and then go to a negatively charged polymer, and you keep repeating that, and they build up this film of zinc oxide on the surface of the material that you're interested in. And when you do that, you significantly reduce the biofilm. So an uncoated on the left, zinc oxide nanoparticle coated on your right, on my left. The zinc oxide nanoparticles, although don't totally remove the bacteria, it is a significant reduction. And when you actually calculate this, and on the left, this is actually a log scale, so you see about a 99% or a 2 log reduction in the amount of bacteria on these. It works for gram-positive Staph aureus, Staph epidermidis. Not so much for E. coli, so we've got some work to do, but it's certainly significant. So what's next? So this is the subject of an R21 that has been reviewed, and we're going to resubmit here in July, but essentially it's to coat these little wafers of material that we're going to do a rabbit implant model. and actually measure how well we can reduce infection in that setting. So the next project, this was originally an RO1 that John Younger had started, and I have since taken over, looking at heat as a way to sort of treat the biofilm when you've already got it in place. And this is based on the idea that some medical devices are so precious, it's really hard to take them out and replace them. LVADs are the most common. I mean, these things, you're going to try to treat through everything you can because replacing this is a real bear. Prosthetic joints, even dialysis catheters, it's a real problem getting these replaced. I think some of you in EC3 have probably waited while patients have had to be, get their dialysis catheters replaced, or even in the ED. It's not a simple thing. So this is based on the idea that heat may sort of augment the effect of the antibiotics on the biofilm. So we had this experiment where we had control cells that are exposed to vancomycin after a biofilm is grown, versus the experiment where we actually added heat, and not a lot of heat. 45 degrees Celsius is not a tremendous amount, but at that thing, 2 hours of it. So I'm going to show you two pictures. On the control, bacteria are green. We put in a dye that'll show you dead bacteria, which is red, and sort of when they mix together, you get this sort of orangey color in there. As you can see, you get a lot of death when you add heat. So both of these are treated with vancomycin. Most of them are alive, but if you add heat, you can do a lot of damage there. So the work that I came in on this, and this is a paper that actually managed to get the cover of Asayo, was to know whether or not we can apply this to a dialysis catheter. Is there a way to actually apply this heat? So thinking about it in terms of a dialysis catheter, this is a computer model we generated saying, if you just pump some hot fluid in there, can you get the catheter warm enough to do this? And so we actually looked at it both on the intravascular portion as well as the subcutaneous tunnel. So when you look at this is sort of the kind of pretty pictures you can generate with computer modeling, is you can actually see the plume of heated flow coming out of this catheter. And we can vary the flow rate, we can vary the temperature, and then we can then vary how much we warm up the blood around it and how hot we get the catheter. More importantly, the subcutaneous tunnel the actual catheter gets really hot, but the tunnel itself doesn't get very warm, so the skin is cooling this off enough. And so we're in a range that we actually think is pretty safe. It's not good enough to do computer modeling. Nobody trusts that stuff. So we actually built sort of a model of this. We're not quite ready to put this in a human yet, but we actually created sort of a superior vena cava with a dialysis catheter, a real dialysis catheter in it, and recreated this. And we can indeed sort of match this up where we kind of set a set point. We want it to be this warm. We turn the pump on and it runs and it warms it up to that amount, brings it back down in whatever kind of protocol we want. And it really matches our computer simulation. All right, so that's sort of moving forward is to try to take that into patients. But the last technology I want to show you guys here is another nanotechnology. This one, tied back to the presentation that Kevin Ward did, this was one of the projects that was funded from the Sepsis Grand Challenge. And the idea here was understanding a better way to diagnose bacteremia. So I get it. This is a really long, kind of complicated title, and I'll try to get at kind of really what the problem is. So here, I think all of us have taking care of this patient that comes in. They're clearly septic, they're febrile, they're altered. You've got all these things, but you don't know the source. Maybe you even figure out the source, but you don't know the bug, and you certainly don't know the antibiotic it's susceptible to. And you're not going to know that information in any time frame that's helpful in a septic patient, because our goal is to administer antibiotics in 1 hour. So you're not going to get any of that information, and that's not very good. Essentially what that is because we use blood cultures. We've already talked about how terrible blood cultures are. They take a long time. 48 hours, 72 hours. They're not very sensitive, they're not very specific, and they don't really find virus or fungi at all. So this is sort of a one-size-fits-all we do for everybody. We throw vanc and cefepime at everybody, or we used to do vanc and zosyn. This leads to antibiotic resistance and opportunistic infections. There's a lot of side effects with these drugs, and sometimes we're under-treating our critically ill patients. So the solution, so this is sort of a typical blood culture, takes 12 to 24 hours to become gram-positive or negative. Another 12 to 24 hours, hey, maybe we know the organism, but we still don't care because you want to know what its speciation is or what its antibiotic sensitivity is. So now you're like 3 days out before you know. This patient could have gone through several iterations of multi-organ system failure by this point. So our idea is to use this nanorod PCR to tell you the organism and the antibiotic resistance genes in basically 3 hours. So the technology is based on using these gold nanorods they're going to attach to a traditional PCR method. And for most folks, they think of PCR for viruses. Bacteria are really hard to find in the blood. They're just too small in number to actually be useful. And so we've come up with a way to actually find those things using traditional PCR, but with this added sort of caveat. So essentially, by attaching the primers of a traditional PCR system to gold nanorods, when you run the PCR reaction, the gold nanorods start to build up these chains, either in long chains or sort of next to each other. And more importantly, when they line up next to each other, you can see this little angle here with a very fancy spectrophotometer called a circular dichroism spectrophotometer. But you can see this at very, very low levels, down to infinitely small, like single bacteria in 10 mils of blood kind of range. And let me explain how good this is compared to regular PCR. So I think most people recognize this. I don't know, does anybody know what lake this is? This is Crystal Lake, Michigan. Yeah, so if you take a penny, let's say 50 pennies, and you drop 50 pennies into Crystal Lake, Michigan, traditional PCR could find that concentration. Our nanorod technology could find 5 pennies in Lake Michigan. Right, so this is a 1,000-fold improvement. So this is what we're talking about, improving this. So as sort of a proof of concept, we've actually done this to sort of differentiate general bacteremia, which is what our 16S ribosomal RNA is there. So it sort of found all the bugs we tested, E. coli, MSSA, MRSA. We also did it to just sort out Staph aureus and then just MRSA. And so that's the output. We don't need to talk about that. That can be converted to a simple number, yes or no. But the best part about this is we get a limited detection of 160 zeptomolar. I just like being able to say zeptomolar. But that's times 10 to the minus 21. So that's very low concentrations. We can do this in 2 hours. And so the next step is really to compare this to blood cultures and sort of move this forward as a technology actually useful for clinical patients. So there's a lot of people to thank here. Most importantly, John Younger, who's my mentor and sort of got many of these things and got me sort of into place to do this. Lots of folks involved in the School of Engineering. And then Dr. Barsan, who was sort of the first one that got me this sort of tool to kind of prove to people that this is a very difficult problem. Funding from many different sources, kind of cobbled together to make this happen. And then happy to answer any questions. So.
Scott, before questions, I just want to say your ability to break down a very complicated question to very simple language is very impressive, and I just want to fund you immediately out of pocket because it seems so compelling when you put it forward.
I'm happy to take the funding.
That TED Talk is like, I think it's a year away. So.